And thank you for the invitation to this fantastic place and the tour we had of the library and the main places and the stored files on all the past members. So I'm going to talk about memory and spatial memory, and more broadly, spatial cognition. And my interest really began by trying to understand memory and how it can go wrong, and to try and bridge this gap that we see between our incredible knowledge now of molecular biology and cell physiology. We know a lot about what happens in cells in dishes, but and we know a lot about how medicines, for example, can affect their behavior, but there's a big gap to understand the behavior in general and memory in particular. And so, as Professor Berthoz alluded to, spatial memory and space is a very powerful tool. And if we look at spatial memory, we can use similar tests of memory for across species. So rodents have to remember where things are, and humans have to remember where things are. And the parts of the brain that we know are required for generating this mnemonic behavior focus on the hippocampus, and the structure is relatively preserved across species. Of course, computational modeling is important to bridge between these scales of individual neurons and molecules and the behavior of the whole system. So I'm going to start with, as Professor Berthoz alluded to, studies by my colleague John O'Keefe, who started to record from the hippocampus of mice or rats as they explored around in a simple environment. So we can see here a cartoon of a rat looking for randomly scattered bits of food in a circular arena. And what the little dots show is a single neuron in the hippocampus firing an action potential, a little pulse of electricity, to its neighboring neurons. And that neuron fires a little action potential, a spike of electricity, every time the rat goes into this place in the box. It doesn't matter which way it's facing. Whenever the rat goes over here, that cell fires a little spike. And so we can draw a firing rate map showing the firing rate of that neuron as a function of the location within the box. And he called those place cells. Together, the population of place cells are telling the rest of the brain where the animal is as it moves around. And a complementary discovery, head direction cells, was made by Jim Rank a little later. These are cells which don't care where the animal is, but they fire according to which way its head is facing. They're like a little compass, so a population of head direction cells tells the rest of the brain which way the animal is facing as it explores its environment. So we can already see the mnemonic properties of these cells in the hippocampus, the place cells. If we record from a population of cells, I'm just showing 3 here, in 2 different environments. What will typically happen is that some cells will fire in one environment, say the circular environment, but not the other one, say the square environment, or vice versa. And once they've learned to distinguish an environment, if you record from the same cells, even 71 days later, if you record from the hippocampus, you see that that learned distinction between environments is preserved. So once the rat has learned that the 2 environments are different, then that information is preserved for a long time. And in terms of computational modeling, what most people think since the work of David Ma is that the neurons which represent the same thing, in this case the same environment, form electrical connections together, synaptic connections, so that if one of them is active, perhaps these ones coloured in red fire in the square environment, then they can reactivate the firing of the others when they're in that environment. And that could constitute memory by, if you activate one part of the memory, it can reactivate the rest of the memory. So we were interested initially in what it is about the environment that makes a place cell fire in a particular location. And by doing experiments where we change the shape and size of the recording box that the animal is exploring around in, you can see that the boundaries of the environment are quite important. So the simplest sort of model we could make from these data is to suggest that a given place cell is receiving inputs from what we called boundary vector cells, or detectors of environmental boundaries, tuned to the allocentric directions, north, south, east, or west, because if you remember, it doesn't matter which way the animal is facing. A place cell will fire when it's in a given location. And maybe a place cell that fires here is getting inputs telling it there's a boundary a short distance to the north, a bit longer distance to the west, further to the east, and even further to the south, so that when you move these two walls apart, you see the firing field of that cell stretching. So we suggested there might be some boundary detectors feeding into the hippocampus that tell place cells when the animal's in the right place for them to fire. So the very simplest model you could think of would be that some number of boundary vector cells tuned to different distances and directions come into a place cell and sum together to make it fire in a given location. For example, if there's something detecting boundaries to the north, then such a cell, if it existed as the rat explored its square box here, would fire at a high rate whenever the rat's in the north end of the box. Another one might be tuned to a boundary to the east and would fire along the east side of the box whenever the animal goes there. And together, they sum together, and if there's some threshold on firing, the place cell will fire in the northeast of that box. So that's the simplest model we could think of. But actually, as well as a model for predicting how place cells will fire as you change the shape and size of an environment, we actually found that there are cells in the, near to the hippocampus which do seem to detect the presence of a boundary in the environment. For example, this shows a single cell, the firing rate maps of that cell, recorded in different environments, either a square one surrounded by a curtain, or a circular environment made of a different material, or even the circular tabletop on which the box had been placed. And what you can see from all of these firing rate maps is that that cell fires whenever there's a boundary to the east of the animal, a short distance, whether it's a curved one or a straight one, or even a drop at the edge of the table rather than a wall. All it's signaling is the presence of a boundary at a certain distance and allocentric direction from the animal. So if we go to humans, I said that I was interested in how human memory works, and we'll look at using spatial memory as a cross-species approach to try and understand what the neurons are doing. We see corresponding deficits in humans. If there's hippocampal damage, they find it very difficult to represent the topographical layout, say, of a computer-generated scene like this. Even if you show them a picture of a scene and then give them a test where they have to remember the arrangement of the mountains, for example, even after a couple of seconds of turning over the page and then they have to choose, then patients with hippocampal damage, particularly on the right, are impaired at this task. Obviously, we know hippocampal damage from famous patients like HM. Their long-term memory is impaired, but it seems that the hippocampus is particularly important for representing topographical layout, or particularly necessary for that, because even if you have to do that for a couple of seconds, if you have hippocampal damage, you find that very difficult. And indeed, this sort of ability is specifically impaired in Alzheimer's disease, and you see bigger impairments in this kind of task for Alzheimer's disease than other kinds of dementia, or indeed than other kinds of memory. So much more recently now, these still not published, or an earlier report in the hippocampus, object vector cells. So we predicted and found that boundary vector cells respond to boundaries. A much smaller portion of neurons in the hippocampus here from this study actually respond to the presence of objects that might be within the environment. And again, with this sort of vector encoding property, they respond to a particular object whenever it's a particular vector, say east in this case, away from the animal, or southwest. And you find the Mosers found similar neurons that respond to the vector away from small discrete objects within the environment. And you can see whenever they move the object to a new location, they get firing in the appropriate location. So we have identified in and around the hippocampus cells that respond to the presence of objects and boundaries in an environment. And we could imagine how populations of those might be used to represent an environment. So what I'm going to show you here is some pictures of a theoretical layout of different neurons where we've put each neuron in a location to represent its tuning to respond to environmental boundaries in this case. This neuron will respond if there's a boundary a long distance away to the north, this one a long distance away from to the south, and these ones short distances, west and east, and so on. So if we were to look down on a population of neurons arranged like this topographically, and say the agent was here in an environment with a boundary of this shape, then if we look down on that population, we'd see a pattern of activity that replicated that environmental layout. So these are the kinds of cells I've been talking about so far in and around the hippocampus, which we know are involved in memory. But of course, perception and action and imagination, in fact, are egocentric. They're not allocentric. So we know that neurons in other parts of the brain, for example, the parietal cortex, which are more closely involved in perception and action and perhaps imagination and imagery, are organized egocentrically. They respond whenever the stimuli a particular distance or direction ahead or left or right of you. So we could imagine the same kind of egocentric boundary vector cells. Now we're going to plot ahead as up rather than north as up. If we look down on a population of topographically arranged egocentric boundary vector cells, we'd see this sort of pattern of activity, because this is the way the animal's facing, and this is therefore the arrangement of the environment, and that's what we'd see. Now, if we're trying to understand memory, we need to know how to go from perception, store something in memory, and indeed during retrieval, take something from memory and generate imagery and remember what the scene around us was like, for example, when we had lunch yesterday. And so what we require is some kind of coordinate transformation. We need to translate from egocentric to allocentric during encoding, and then from allocentric back to egocentric during generation of action or imagery. Now, there's people who studied the parietal cortex made originally Anderson's group, but also Duhamel, and theoreticians like Alex Pouget and Sophie Deneuve have thought about this problem a lot. And in the parietal lobe, you find neurons called gain field neurons that respond both to the egocentric location of, say, a visual stimulus, but the firing rate is modulated also, for example, by the direction of the head in the world. And these gain field neurons, responding to both kinds of information, are perfectly adapted for doing this translation. In fact, you can wire up, if you see something on the right and you're facing north, so it's actually to your east, then if you activate these cells and the corresponding conjunctive gain field neuron, you could wire them all together. And if you do that for all combinations, you end up with a system that can translate effortlessly from left, right, or ahead to north, south, east, and west, as long as it knows which way you're facing, and can translate back again from allocentric to egocentric. If you know you're facing north, you know things to your east should be on your right. So with this kind of circuit, which we think is in parietal, perhaps medial parietal or lateral parietal areas, then we could start to try and make a sort of model of, a functional model of human memory. So here's the hippocampus, sort of deep within the human brain, seen from the inside out. And here we think boundary vector cells and perhaps object vector cells should be in neocortical areas nearby, and some cells which represent the identity of the things you're looking at in perirhinal cortex. And then we have the parietal areas, which we think are egocentric, and we need to make this egocentric to allocentric transformation informed by our head direction cells, which tell us which way we're facing. So this could be a sort of layout of how memory is organized, and these connections between all of these regions here in the medial temporal lobe could allow memory in the sense of pattern completion. If I can remember where I was, then I can perhaps reactivate which objects and boundaries were around me, and therefore generate a scene to imagine when I'm trying to recall what happened. And so what I'm going to show you is a computer simulation of these populations of topographically organized cells, boundary vector cells or object vector cells, head direction cells, place cells, and here the egocentric parietal cortex cells, the egocentric versions of object and boundary vector cells, identity neurons. And this thing here is the agent, this little triangle. And what we're going to see is the agent moving around a familiar environment, so the boundary vector cells and place cells have all been connected together already to represent the configuration of this environment. And as the agent walks around here, it's going to encounter a novel object and learn some new associations between cells encoding this object and the place cells in that location and the boundary vector cells. And then we're going to see how those associations can retrieve the memory and enable it to form a imagined scene of the event in which it encountered this new object. So here we go. These are place cells. As the agent starts to move, you can see the bump of activity on the place cell sheet is tracking its location. The head direction cells are tracking its orientation, and the sensory input is coming in through this parietal system, being translated and activating object vector cells and boundary vector cells. These cells are encoding the identities of these boundaries and the identity of the object. So it's now encountered this new object, perhaps it's some keys. The parietal neuron is active, that represents keys, and the activity of these object vector cells for this object, those cells can now be wired together with the place cells for that location and the boundary vector cells for the surrounding scene. We think long-term potentiation of synaptic connections will do this. And so now, having encoded this information, the agent can continue on its way, and having encoded that memory in those changed synaptic connection strengths, it goes about its business now, still receiving perceptual input in an egocentric frame, driving boundary vector cells that drive place cells to fire. But now perhaps it gets a signal to do memory recall. Perhaps its partner says, "Where did I leave my keys?" And so by activating the neuron representing keys, because of the learned connections back to place cells encoding the location of the object and the boundary vector cells encoding the scene at that point, it should be able to drive top-down now from this allocentric stored representation back into imagery, an egocentric representation of the scene when it first encountered that object. So we can see now the activity is spread from the identity neuron back to the place cells, so it's now representing the location where it was when it encoded this object. And that activity is spread to reactivate all of the boundary vector cells for the whole scene around it, including the ones that are out of view. But in memory, because it's a familiar environment, it knows where everything is around this location. And then being translated from allocentric to egocentric through this parietal circuit, which I've not shown, it can generate a viewpoint on a scene, a spatial scene, of its encounter, where the object is right in front of it, and this is the room around it at that point. And so what we've done is make a neural level model of what happens when you have an encounter with something, and then you have to remember what happened and bring it to mind and imagine it. So that's a sort of static view of memory, if you like. Sensory perceptual inputs can drive representations of location and indeed of head direction. And we can begin to piece all these spatial representations together to understand how memory might work in the sense of encountering something and storing it in long-term memory where your orientation doesn't matter. You've remembered the layout of the environment. It doesn't matter which way you're facing it. You've remembered where things are arranged relative to other things within the environment, and using that to generate an egocentric representation, so you can imagine what's ahead of you and left and right of you and so on. So now, in 2005, the Moses, who Professor Bertos also mentioned, discovered grid cells. And so here's a similar cartoon of a grid cell. So in the entorhinal cortex, the medial entorhinal cortex that projects into the hippocampus here, if you record individual neurons, you can see this rat massively speeded up in this cartoon, exploring around in a large square box. And each dot now marks the location when a single neuron fires an action potential. And you can see that the same neuron fires action potentials whenever the animal goes into any of a series of locations, all arranged on a nice regular triangular lattice. So if we make a firing rate map of firing rate as a function of location within the box, we see this regular triangular grid of activity locations. If you record from neighboring neurons, neighboring grid cells, you see that they've got similar grid-like patterns, but they're just slightly offset from each other. So they have the same orientation of grid, but they're slightly offset relative to their neighbors. Indeed, the scale changes as you go further down in the brain. So it's thought that because each cell fires in this rather rigid pattern, whatever the environment is, you see similar firing. If I put this rat now in a different, say, a circular environment, it wouldn't care. The grid cell would still fire like this, and its neighbors would still fire similarly. So wherever the animal is, if it's sort of heading north, it knows that activity can pass from the red cell to the green cell and then to the blue cell. And so people think that maybe this is a system for updating where you think you are based on your own self-motion. It could be intrinsically wired to keep track of where you are, irrespective of the environment around you, driven by your own self-motion signals. So what we're coming to now is a sort of picture where place cells might be predominantly or at least significantly driven by environmental information, like boundary vector cells or object vector cells, to fire in a given location in a given environment. And grid cells can update their pattern of activity according to self-motion cues. But actually, anatomically, both kinds of cells connect to each other. And so it makes sense, computationally, what we think is that the place cells will form connections with grid cells which fire in the same place. And the connection from place cells to grid cells is useful because the grid cell will fire with exactly the same grid-like pattern day after day, which could not be maintained by self-motion. When you go away and come back again, you need to anchor your grid-like firing pattern to something in the environment, which could be a place cell which is being driven by sensory inputs. And equally, in the return direction, it means that your self-motion can update your sense of where you are, even if you're not getting very reliable sensory inputs, or you've closed your eyes, or you're not paying attention. And so together, these two kinds of cells can combine to make a sort of optimal combination of environmental and self-motion information to keep track of where you are as you move around. So going back to humans, we would like to see if we can find this kind of grid-like signal in humans. And so we can get people to play video games in the fMRI scanner where we can probe their spatial memory. So one of these games would look like this. You would explore, in this case, a circular environment, and then your task would be to go around and find objects like this little duck. And your task is to remember where it's located, so that if you're shown it again and put back into the environment, you can find your way back to where it was, where you found it, and press a button to indicate your memory for its location. So if we have people doing that task with many, many objects over many trials in an MRI scanner, here's the path somebody took during one of these experiments seen from above. Because the grid-like firing patterns of all of the grid cells are aligned, they share these same 6 axes of symmetry. Even the grid cells that are further apart and might have different scales. We think there'll be a different dynamics of firing if you're running along these axes of symmetry, shown in orange, compared to between them, these directions shown in grey. So we can look in the scanner for fMRI signal that varies as a function of running direction and goes up and down every 60 degrees. And that would be an indication of some sort of spatial representation that had this 6-fold symmetry. Okay. So when we do that, we find activity in right entorhinal cortex corresponding to rodents, but also in other areas throughout the autobiographical memory system, suggesting that perhaps these grid cells that allow self-motion to update our sense of location could be used to drive movement of the viewpoint you use to generate imagery in the recall of events in the model that I just showed you. And now, just to sort of wrap up, I want to move back from space again. We use space to understand what these neurons are doing and how they might work together. But actually, we are interested in episodic memory more generally. And if you record in human hippocampus, you typically see neurons. You do see place cells when people play this kind of video game, but you also see cells that are tuned to respond, for example, to famous people, the famous Jennifer Aniston cell, for example. Or in Queen Square in London, we have cells that respond to Mick Jagger or Gary Lineker, or even Greg's the bakery. It's a very cheap bakers that you can go to. And in a fantastic experiment by Tim Behrens' group, he thought about the navigation experiment we had just done, showing this six-fold symmetry in fMRI activity with movement direction. And they trained people to navigate in an abstract space of detecting bird types from bird silhouettes in which the length of the neck or legs could vary. And while people were in the scanner watching these trajectories of bird silhouettes, they did the same analysis that we had done and found the same six-fold symmetry in the same regions, indicating that perhaps there's a grid-like representation of abstract conceptual knowledge, not just space. So now, finally, we might think that although we've studied it in space, what we're seeing is that place cells are representing concepts, if you like, more generally, in a sort of locally tuned way, perhaps driven by all sorts of feature vector cells that tell it where it is in this conceptual space, and that grid cells are kind of, well, they would be a very compact way of modeling the transition structure of this state space, a bit like a Fourier representation, a way in which you could use this representation to generate the vectors between any two different concepts. And so this would be an interesting way to organize conceptual knowledge, including all vector representations between concepts, and that would be a very powerful thing. So perhaps that's what we're seeing more generally, and I'm running out of time, so I'll just say very briefly, because we've got a neural level model of how at least spatial memory and imagery works, we could use it to try and investigate what goes wrong, for example, if there's very strong negative emotional affective content, and how things like post-traumatic stress disorder might develop, but I don't have time to talk about that. So to conclude, I think by studying spatial representations in these freely moving rodents and also in humans, we've got a mechanistic understanding of how egocentric and allocentric representations interact with each other and how that relates to spatial memory. And perhaps we can use that to begin to understand episodic memory and the storage of conceptual knowledge, and hopefully eventually how they can fail in different neurological and psychiatric disorders. I should just thank Andrzej Bikansky was the person who programmed the model that I just showed you, and various forms of funding. Thank you very much.
Thank you very much. We may have time for one or two brief questions. Would like to ask a question? Yves Agile and Jean-Pierre Changeux, and this will be...
I'm here. Well, in most instances, in humans at least, or I think it's the same in animals, this type of behavior are automatic, right? The brain regions we are supposed to be involved in automatic behaviors are the basal ganglia. So what about the connection between the cerebral cortex and the basal ganglia?
Well, this is a great question for, I think, Laura Rondi-Reig is going to talk about this exactly. But in terms of...
Thank you, that's great. If she answers, that's okay.
Well, I'm guessing, but I've known her for a while, I'm guessing she will. Yeah. I would say these behaviors, effortful autobiographical memory where you deliberately bring something to mind is not really automatic. But Laura will say more about this.
Okay, beautiful presentation. HM, and according to some surgeons from UCSD where I'm working, does not seem to have any problem of environment location. And these surgeons in UCSD have also confirmed this fact. So I understand that in your scheme you have recently amplified the system to other places in the brain. So do you think that the extension of the rat system to the human is just not appropriate, or and if there is something different in humans compared to rats?
Well, the first point I want to make, it goes back to this question. So I've only described one brain system, and there are many others, including the basal ganglia, many of which can support various aspects of spatial behavior. Now, what you're talking about is still controversial in terms of exactly what it is that HM could remember about his spatial location. So when they took him back to where he had lived 20 years before the operation, he was able to perform tasks, spatial tasks. But one suggestion is that he had been very familiar with this location where he'd grown up, and it had become automatized, and perhaps basal ganglia or striatum and maybe parietal cortex was enabling him to do use the kind of representation which Law will talk about, which is difficult to distinguish from a fully cognitive representation where you can bring things to mind. But there's a general controversy here about whether information in the hippocampus is eventually transferred to neocortex, and therefore, if the information was learned long enough before the surgery or the epilepsy, which probably started to impair his hippocampus before the surgery, if it was long enough ago, then neocortex can support behavior even without the hippocampus. And that's still controversial. So most people suggest that when HM was asked to describe the incidents from his past, which he could indeed describe, that he always used exactly the same form of words, and this was like an overlearned, rather automatic response, rather than being able to bring it to mind and flexibly investigate different viewpoints within it. And so it's a good point, but it's still controversial, and I obviously side with the people that think there are some aspects of spatial memory and episodic memory and imagination which HM could not have done if he'd been tested carefully enough. But he's no longer here, and the different patients, different people argue about. So obviously humans are different to rodents, to answer your question. I think we can learn a lot about the neural mechanisms nonetheless, because we can do more invasive experiments in rodents. But I think spatial memory is rather similar in rodents and humans. Humans then have a lot of other kinds of memory which rodents don't. Yes, yes. So the Fried Kahana group, yes, epilepsy patients with electrodes playing this kind of video game. They're not as common as Jennifer Aniston type cells, but they are cells that encode location. And in one study, they had people find different objects in different locations in a video game. And when they had to remember the objects, they showed incidental reactivation of the place cell that fired at that location. So what evidence there is confirmatory, but there's a lot of other things going on in the human hippocampus which are not present in the rat. I would agree with that.